Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientist monthly webinar series. I'm Dr. Jeff Borenstein, BBRF's President and CEO. Today, Dr. Laura Berner will present Timing is Everything. Repeated neurocognitive assessments can help us understand symptom patterns in bulimia nervosa. BBRF is the world's largest private funder of mental health research grants, supporting transformative discoveries in order to develop improved treatments, cures, and methods of prevention. The high quality of the research we fund is made possible by the BBRF Scientific Council. This group of 195 prominent mental health researchers reviews each grant application and selects the most promising ideas with the greatest potential to lead to breakthroughs. The Scientific Council guides the foundation to fund creative and impactful basic, translational, and clinical research relevant to the whole spectrum of mental health. One reason that research funded by BBRF has such great impact is because we do not limit our focus to one illness or condition. Since 1987, the foundation has awarded $450 million to fund more than 6,500 research grants around the world and across a broad spectrum of brain illnesses, including addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, post-traumatic stress, schizophrenia, as well as suicide prevention. Today, I'm delighted to introduce Dr. Laura Berner. Dr. Berner is an associate professor of psychiatry at the Icahn School of Medicine at Mount Sinai. She was a 2020 Young Investigator. Our webinar will begin with Dr. Berner's presentation, which will then be followed by Q&A. To submit your questions, please use the Questions tab on the control panel on your screen. Please feel free to submit your questions at any time. Following the presentation, I'll ask as many as is allotted by the time. And now I'm pleased to introduce Dr. Berner. Laura, the floor is yours.
Great. Well, thank you so much for inviting me back to present in this series. I'm really excited to tell you about some more of our recent investigations focused on developing a nuanced understanding of bulimia nervosa using repeated measures assessments in different states. Before I keep going, I just want to confirm you can see my screen okay?
Yes.
Yes.
Okay, fantastic. All right, so let's start with defining bulimia nervosa. What you see on the screen are circles representing our major specified eating disorder diagnoses according to DSM-5. And you can see that bulimia nervosa, which is represented by the yellow circle, really lies at the center of things because it's characterized by two key behavioral features that cut across our eating disorder diagnoses. The first of these behavioral features is recurrent episodes of binge eating. This is defined by eating a large amount of food in a discrete period of time, but it's different from just overeating, and even overeating that someone might feel distressed about or regretful of. It's critically defined by experiencing a sense of loss of control during the episode. Folks often describe this feeling like a ball rolling down the hill or feeling like it's hard to stop once one has started. The second cross-cutting behavioral feature of bulimia nervosa is compensatory behaviors. A lot of people think first of self-induced vomiting when they think of bulimia nervosa, but the disorder can also include laxative, diuretic, or medication misuse, excessive exercise, or even fasting behavior. So self-induced vomiting isn't necessarily required for this diagnosis, but any one of these behaviors can be occurring regularly as an attempt to compensate for binge eating episodes. Bulimia nervosa is the second most common specified eating disorder diagnosis after binge eating disorder, and it affects around 2% of the population. But the symptoms of bulimia nervosa are actually more common than you might think. For example, 1 in 5 women say that they've had episodes of binge eating, and 1 in 3 have made themselves vomit or taken pills to control their shape or weight. And unfortunately, these rates are even higher among folks who are transgender. In addition, historically, it was often thought that this disorder predominantly affected white women, but more recent research shows that individuals from minoritized populations, particularly Black or African American and Latinx individuals, experience similar or even higher rates of bulimia nervosa. Folks from Asian American backgrounds also report significant rates of bulimia nervosa, but disparities in diagnosis and treatment might exist because of cultural stigmas, differences in healthcare access, and biases in the medical community, potentially leading to underdiagnosis in these populations. Research indicates that binge eating and self-induced vomiting are between 46 and 72% heritable. So along with our brain imaging research, this really highlights that these behaviors are not just about societal pressure to look a certain way or to fit into a certain size and about choices related to those desires, but that they have powerful underlying biological components that are driving them. This disorder is also associated with significant impairment. 78% of individuals with the disorder report at least moderate disability, and it's linked to a very high 4% mortality rate. Outcome of folks with this disorder is variable. The disorder typically persists for several years, and about a quarter of patients end up, unfortunately, following a chronic course of illness. Importantly, over 60% of our patients who receive current first-line treatments for bulimia nervosa remain symptomatic, which really highlights an urgent need to identify the mechanisms that maintain these symptoms so that we can develop better and more targeted interventions for them. This is the major mission of my research program as both a neuroscientist and a clinical psychologist. My research specifically focuses in on the complex process of engaging self-control and how and why it is that our patients seem to get stuck in extremes of control, out of control binge eating and purging, often prolonged over-controlled dietary restriction between these episodes, and even volatile relationships. Part of what has limited this understanding to date, I would argue, is that most work in bulimia nervosa has been focused on potential stable or trait-like differences in the neurocognitive processes in individuals with the disorder. For example, a lot of our models of bulimia nervosa rest on the assumption that individuals with this disorder have consistently low behavioral, attentional, and emotional control. But in our recent work, we started to think about how bulimia nervosa, almost by definition, doesn't just involve constant problems exerting control. Sometimes these folks are very rigid and perfectionistic and seem to engage too much control. So perhaps bulimia nervosa is defined by problems adaptively exerting control to match the current situation or the needs of the particular state that the person is in. So the work that I'll tell you about today has really focused in on trying to identify which specific elements of the control process might be going awry and in what specific contexts. So what do I mean by elements of the control process? Well, I think often when people think about exerting control, they think about it in a simple binary red light or green light kind of way. You either go or you stop. You exert control or you don't. But think about all the ways that you would need to exert control if you were driving on a dark, windy mountain road at night that was unfamiliar to you, that had a lot of dynamic states and twists and turns. Yes, you would need to be able to successfully press the brake or not, but you also need what maps and signs and app alerts like this can help us do, to build up a model in our brain of what the road ahead looks like so we can anticipate what's coming up next. We can estimate how expensive a decision to take one route will be in terms of time, tolls, or potential nausea that we'll experience on hairpin turns, and incorporate information about what might have just changed in the environment so that we can adjust our control accordingly. Data from cognitive neuroscience suggests that we can think of applying control adaptively in our everyday lives as a similar process of gathering sequential data so that we can update our expectations about control. We can decide whether exerting control is even worthwhile, and in turn, we can adjust our behavior. Certain pieces of this dynamic and complex process of adjusting control might go awry at certain times in bulimia nervosa. And these pieces may be more precise targets for treatment. So our work has used brain imaging, novel tasks, and mathematical models of decision-making and behavior to try to pinpoint them. Today, I want to tell you about some of our research that's designed to address the question of how and when self-control processes go awry by asking the same participants with bulimia nervosa to complete tasks in different states. First, I'll show you results from 2 studies that really focus in on problems related to adaptively engaging control when one's internal context changes, specifically after the state change that's introduced by eating. And then I'll show you results suggesting that individuals with bulimia nervosa have difficulties adjusting control after non-eating related external context changes occur. So all the studies that I'm going to show you today focus in on problems related to control when there's been some sort of a state change. And maybe the most obvious state change that's relevant to people with eating disorders and that we all go through every day, multiple times a day, is when we move from a hungry, fasted state to a satiated, fed state. We've probably all seen the long-running Snickers ad campaign built on the idea that if you don't have an eating disorder, going for too long without eating can make you feel pretty dysregulated and out of control. And there are actually data to back up this idea. Fasting deactivates a part of our brain that's critical for self-control, the lateral prefrontal cortex, and it impairs inhibition. On the other hand, eating can increase lateral prefrontal cortical activation and enhance inhibition. And this makes pretty good evolutionary sense, since we need to be more bold and motivated to explore and disinhibited when we haven't eaten for a while, and we need to stay put and focused to use our energy wisely after we've eaten something. However, in people with bulimia nervosa, we know very little about how this system functions or malfunctions. But it seems particularly important as folks with bulimia nervosa fluctuate between these extremes of out-of-control binge eating and compensatory behavior on one end of the control spectrum to dietary restriction or prolonged fasting behavior between these episodes on the other end of the control spectrum. So the first study that I want to tell you about today focuses on how one specific component of the self-control process might go awry when individuals with bulimia nervosa are in fasted or fed states. The study was built on recent findings from mathematical models of behavior in combination with brain imaging, suggesting that people's brains encode a comparison between our control-related predictions and our actual experience of how much control was needed or whether it was needed. These comparisons or prediction errors are critical for updating our predictions about control and for helping us adapt our behavior. And they're encoded in parts of the brain called the dorsal striatum and the lateral prefrontal cortex, or dorsal frontostriatal circuits. We specifically hypothesized that if individuals with bulimia nervosa have trouble with this process of flexibly adjusting control or engaging the regions of the brain that support this flexible adjustment of control when internal contingencies change, specifically after eating or fasting, these individuals could get stuck in extreme versions of both of these behaviors. To begin to test this question, we conducted a model-based analysis of functional brain imaging data collected at UCSD from healthy controls and women with a history of bulimia nervosa. So these women were remitted from the disorder. They hadn't had any symptoms of the disorder for at least a year, but they had a history of meeting full criteria for the disorder. All of our participants stayed for 72 hours in a controlled environment and completed 2 scans over 24 hours in a counterbalanced order. That means some people completed one type of scan first and some people completed the other type of scan first. These 2 scans occurred, one after a 16-hour fast and the other type of scan occurred after a standardized breakfast. At both of these scans, participants completed a stop signal task. This is a common paradigm used for studying inhibitory control. Participants are instructed to press a button in response to a go signal unless a stop signal occurs at some point after that go signal. And in this version of the task that we used, the stop signal was a beep, and it was presented with a delay after the onset of the go stimulus. The longer the delay, the harder it becomes to inhibit responding because a response might have already been initiated to that go signal. This delay varied over the course of the task, completely independent of participants' task performance. In other words, the likelihood of a stop signal and the stop signal delay itself were completely unpredictable to participants. So if we remember all those things that you needed to do to successfully navigate a dark, windy, unfamiliar road at night, Bayesian ideal observer models, or mathematical models, provide a computational framework to help us precisely quantify some of these things. In other words, they help us quantify how the brain uses and updates expectations about control based on context and experience to optimize our behavior. We applied a now well-characterized dynamic belief model, which is a particular hidden Markov model developed by our collaborator Angela Yu and now used in 5 studies of substance use disorders, to understand adaptive inhibitory control on this stop signal task. The model assumes that the estimated likelihood that a stop signal is coming, or p-stop, is critical for selecting the appropriate action policy or strategy, just as the estimated probability that a hairpin turn on a dark road is coming is critical for us deciding whether or not to slow down. In this model, p-stop, or this estimated probability that a given trial will be a stop trial, is essentially the mean of a predictive prior distribution, or an expectation distribution, and it gets continually updated in each moment using a mixture of these expectations and your experiences and Bayes' rule. Instead of walking you through this kind of scary looking equation on the screen and all the math that goes into this model, I'm going to walk you through what its application to a couple trials of this task look like. So let's say that in a given moment, you think there's a 30% chance that you're going to need to inhibit your behavior. That means you think in this task that there's a 30% chance that this trial is going to be a stop trial. It turns out that it was actually a go trial. So the difference between your experience and that expectation or prediction error has a magnitude of 0.3, and it's negatively signed because you expected that you were going to need to inhibit your behavior and you didn't need to. This prediction error helps inform the updating of your expectations for the next trial, or your p-stop estimate for the next trial. Now you can see it's gone down to 20%. It turns out, though, that you did need to exert control in that moment. This was a big surprise. So you have a prediction error with a magnitude of 0.8, and it's positively signed here because you needed to exert control when you weren't really necessarily expecting to have to. That prediction error again helps update your expectations for the next moment, and now you think that there is a 25% likelihood that you're going to need to exert control. So you can see how the difference between your expectation and your experience, which is thought of as a kind of Bayesian prediction error, encodes the magnitude of surprise, or the degree of discrepancy between your internal predictive model of the world that's captured by p-stop and the outcome that you actually experience. This mathematical approach permits modeling of the trial-by-trial updating of our predictions based on control prediction errors of forthcoming control-related demands. And given the sequences of observed p-stop and go trials and the settings for other parameters in the model, we can compute a sequence of p-stop probabilities and prediction errors for the sequence of stimuli that each subject experiences, and then use them as regressors in our fMRI data analysis. So this allows us to essentially see where in the brain, if we look across the whole brain, are these particular parameters where, for example, are prediction errors being encoded in the brain and tracked in the brain. We focused our fMRI analyses on these Bayesian prediction errors because they've really been shown to be key to understanding another group that struggles to control their behavior, substance use disorder populations. So what did we find at a behavioral level on this task? Well, as you can see in these graphs showing the remitted bulimia nervosa group in blue and healthy controls in red, there were no group differences or group by state interactions in errors of commission or omission. In addition, there were no group differences or state differences or group by state interactions for stop signal reaction time. And as predicted by our Bayesian model that we applied to the data, there was a significant positive relationship between individuals' trial-by-trial p-stop model estimates and go reaction time in each group. So with higher p-stop estimates, go reaction time slows down to help promote correct stopping on a stop trial. So this makes sense. The more likely you think it is that you're going to need to inhibit your behavior in the moment, the more you should slow down so that you have a greater chance of successfully inhibiting your behavior in that moment. This relationship between p-stop estimates and go reaction time, which is sometimes called the sequential effect, didn't differ across states or between groups, and there was no group by state by p-stop interaction for go reaction time. This lack of behavioral differences might not be surprising, given that the clinical group in this study, if you remember, were women who were remitted from bulimia nervosa and had not engaged in any bulimic behaviors or symptoms of the disorder for at least a year when we studied them. However, although our behavioral analyses didn't detect differences between our groups or interactions with state, our model-based brain imaging analyses did. In the left dorsal caudate, we detected a group by state interaction for activation encoding unsigned prediction errors. Healthy women had increased prediction error dependent activation in the left dorsal caudate in the fed state, which is shown on the right, compared to the fasted state shown on the left. And this is really consistent with the notion of the brain being in an optimized state for engaging control after eating. However, the inverse relationship was observed for women remitted from bulimia nervosa, who showed increased activation tracking these prediction errors in a fasted state and attenuated prediction error dependent activation after eating. Interestingly, adults with methamphetamine dependence show similarly reduced activation in the left dorsal caudate tracking unsigned prediction errors on the stop signal task. So this suggests that blunted caudate tracking of control-related surprises might be a transdiagnostic contributor to behavioral symptoms like binge eating and substance use that both feel out of control. In addition, we found that this blunted signal tracking the unsigned prediction error in the fed state in women with a history of bulimia nervosa was linked to more frequent past binge eating and self-induced vomiting. And the only other study to our knowledge to apply model-based fMRI to bulimia nervosa similarly found reduced reward prediction error signals for taste of sucrose. These were not in the caudate, but in the insula and ventral striatum, and this reduced neural signal for prediction error tracking was similarly associated with more frequent weekly binge purge episodes in women who had current symptomatic bulimia nervosa. Therefore, striatal tracking and integration of the information about the discrepancy or mismatch between our expectations and experience in both reward and control-related domains may be altered in association with bulimia nervosa and might help to explain symptoms. Our state-specific findings suggest that this group might be less sensitive to surprises in the realm of control, specifically after the state change introduced by eating. And this could result in difficulty switching their control-related strategy in the fed state, making it feel hard to stop eating once eating has started, and promoting additional subsequent out-of-control behaviors like self-induced vomiting. This is certainly consistent with at least some patients' descriptions of what loss of control eating or binge eating feels like. For example, I start off by treating myself to one thing, then I go blind and all I know is food. Or I try to be better about eating. I know I have to eat, and then it turns into loss of control eating. The next study that I want to tell you about, which is also focused on the state change introduced by eating, focuses in on another subcomponent of the control process, a cost-benefit calculation. Specifically, individuals need to weigh the costly cognitive effort of trying to exert control against its benefits, to determine if it's even worth it to try to control their behavior. In contrast to the dorsal circuitry that we just saw, this computation is encoded in ventral frontostriatal circuits, primarily involving the ventral striatum, the anterior cingulate, and the ventromedial prefrontal cortex. We posited that this value-based decision-making about control might also fail to adjust appropriately to the current context in individuals with bulimia nervosa. Our collaborator, Andrew Westbrook, developed a now well-validated task to measure how individuals weigh the costs versus the benefits of the effort associated with cognitive control using a discounting procedure. In this task, participants first experience 4 levels of an NBAC task. This is a cognitive control task that instructs participants to respond, depending on the level of difficulty, only to stimuli that they last saw either 1, 2, 3, or at the hardest level, 4 trials before. After completing these 4 levels of the NBAC task and really getting familiar with how difficult each level is, participants then made choices between either having to repeat a higher level of the NBAC task, which is a harder task for more money, or a lower, easier level for less money. These offers were presented to participants until the point at which a person reaches indifference is identified, and this represents their estimated subjective value of cognitive effort. Some recent data using this task suggests that part of the Snickers ad campaign effect of eating on self-regulation in healthy people may be related to the dopamine release that's associated with food ingestion. Pharmacological agents like methylphenidate that induce dopamine release in brain circuits that connect the ventral striatum to the ventral prefrontal cortex can increase the subjective value of cognitive control-related effort. And they can also bias individuals toward the benefits versus the costs of this effort. It turns out that eating releases dopamine in those same areas of the brain. So given these findings, we hypothesized that for people without eating disorders, trying to control one's behavior just might seem more worthwhile after eating something, but that this state-specific cost-benefit computation is altered in people with bulimia, such that the cost of exerting control would abnormally increase or remain abnormally high after eating. To test that question, in my BBRF NARSAD-funded project, we have women with bulimia nervosa and matched healthy controls complete the task after a 16-hour fast, and then again after a standardized meal. Preliminary behavioral results from this task suggest that eating does in fact differentially affect effort-cost computations among individuals with bulimia nervosa. After eating, much like after drugs like methylphenidate, healthy women shown here in this graph on the right in red demonstrated an increase in the subjective value of control-related effort. So this was in line with what we hypothesized. However, in line with what we hypothesized, women with bulimia nervosa, who are shown in the graph at right in blue, did not show this increase in subjective value after eating. Compared to healthy women in a fed relative to fasted state, women with bulimia nervosa show a lower subjective value of cognitive effort. In other words, they're less willing to exert cognitive effort because, as indicated by this task, they find it to be of lower value, or to view it from the flipped perspective, they view it to be more costly than healthy controls do after eating. We wanted to test whether participants with bulimia nervosa also performed worse on the cognitive control task used here, which was the NBAC task, in the fed state. If they did perform worse after eating, their higher effort costs after eating would just be an accurate shift that would align with their poorer performance in this state. However, we found that there were no differences by group or state in task performance, suggesting that the state-specific changes that we saw were solely in the valuation of cognitive effort. Therefore, the results suggest that women with bulimia nervosa may have inaccurately inflated subjective costs of cognitive effort or overestimated costs of cognitive effort after eating. These relatively higher costs of control in a fed compared to fasted state might lead individuals with bulimia nervosa to conclude that trying to exert control after eating starts just isn't worth the cognitive effort. And consistent with this idea, we found that state-specific cognitive effort cost estimates scaled with symptoms. Women with bulimia nervosa who had the lowest subjective values of cognitive effort across NBAC levels after eating, which is represented by an area under the curve calculation, had more frequent binge eating episodes in the last 3 months. And although we didn't see group differences in the fasted state, women with bulimia nervosa who had the highest subjective values of cognitive control-related effort in the fasted state reported more severe dietary restriction between binge purge episodes. So these associations with symptom measures suggest that state-specific alterations in effort cost computations could maintain binge eating episodes that then alternate with periods of dietary restriction. Again, these findings are in line with at least some patients' descriptions of what loss of control eating or binge eating can feel like. For example, I already started, I'll just go all the way. I feel like if I tried hard, I could stop, but I just don't care. Or I already blew it, I'll go for the whole thing. These state-specific data so far support our hypothesized model of bulimia nervosa that could help explain the extreme eating behaviors that we see and that we're now testing in a larger R01 study. Specifically, we hypothesize that when these individuals are in a fasted state, increased dorsal frontostriatal circuit activation encoding control-related prediction errors, and increased ventral frontostriatal circuit activation encoding control-related valuation promote restricted intake that continues, and ultimately this becomes a self-perpetuating cycle where being in the fasted state alters these computations and ultimately promotes continued fasting. However, once individuals do initiate eating and they move into a fed state, this model proposes that decreased dorsal frontostriatal circuit activation, tracking those prediction errors, and decreased ventral frontostriatal circuit activation encoding that control-related valuation process could promote eating that becomes excessive and out of control, subsequent self-induced vomiting and laxative or diuretic misuse that can also feel pretty out of control to patients, and that ultimately return individuals to the fasted state. So this could explain how individuals vacillate or oscillate between these two extreme behaviors. And we're testing these ideas in individuals with current symptoms of bulimia nervosa in the scanner using the tasks that I've showed you so far, but also in the wild using mobile phone-based assessments that allow us to assess how individuals in each of these states might more naturalistically be affected by these states. If our findings end up being replicated in larger samples that exclusively include individuals with current symptoms of the disorder, our findings may also have direct implications for the learning that's required to control and change behavior in treatment. Specifically, rather than first focusing on and attempting to eliminate restriction, as we do in our first-line cognitive behavioral therapy for bulimia nervosa, perhaps it would be useful to focus on delivering interventions like neurocognitive training, non-invasive brain stimulation, or exposures and skills practice while participants are consuming food or while they're in a fed state. And perhaps these interventions should focus in on enhanced tracking of control-related surprises, or decreasing the perceived costs or boosting the perceived benefits of expending effort on cognitive control. So I've shown you that bulimia nervosa and more severe symptoms are associated with abnormal functioning within self-control circuits after eating, but those findings can't necessarily explain all the symptoms, including non-eating specific problems with control that we see in folks with bulimia nervosa. In addition, our findings so far suggest that people with bulimia nervosa do not adapt their control processes to internal changes in the same way that people without eating disorders do, but we don't know whether these processes are also sticky or show alterations after external changes occur. To begin to address those gaps, we've done some work examining how individuals with bulimia nervosa differentially adjust or fail to adjust to changes in contingencies more broadly. We were specifically interested in this notion because of how it might help us understand persistent treatment-resistant symptoms of bulimia nervosa. One possibility that we wanted to test is that an altered ability to engage goal-directed control to stop engaging in behaviors that no longer lead to valuable outcomes plays a role in the maintenance of bulimic symptoms. For example, we often see that people with bulimia nervosa continue binge eating and purging despite a reduction in their once reinforcing effects and negative consequences like guilt and shame, health risks, and affective instability. This could reflect a failure to adjust to outcome devaluation. But we also know that behavioral adjustments are required after we get new information about the predictive value of the cue or the stimulus that precedes the behavior. An impaired ability to adjust behavior when the values of these stimuli change could result in automatic bulimic behaviors in response to particular cues. For example, the sight or smell of binge foods or strong negative emotional experiences. And these could persist despite efforts to respond in different ways to those cues. For example, efforts to use an adaptive emotion regulation skill instead. These difficulties could reflect a failure to adjust to stimulus devaluation. So in this next study, we investigated the ability to adapt to temporary devaluation of either stimuli or outcomes in women with bulimia nervosa and group-matched healthy controls. Here, we didn't explicitly control for participants' fasted or fed state, but both groups ate approximately 6 hours before completing the task, and there were no group differences in the time since last food consumption when both groups completed it. We used a task that was originally adapted by our collaborator Sana DeWitt from rodent models to measure outcome devaluation, and in some versions of the task, stimulus devaluation. It's been used to study various clinical populations, including OCD and alcohol use disorder, and other variations of the task have been used to study cocaine addiction and nicotine addiction. The task has 4 stages and it's pretty complex, so I'm going to walk you through it in a couple of different ways. In the first stage of the task, participants learn that they need to make a particular response to specific stimuli, and that if they make a correct response to that stimulus, it will lead to a specific outcome. The second stage tests whether participants learned which responses led to which outcomes. That's because the first phase of the task, even though participants are learning about associations between stimuli and responses and outcomes, the first phase of the task only assesses knowledge about stimulus-response pairs. So the second phase of the task is really designed to assess knowledge about response-outcome pairs. And the third and fourth stages of the task introduce the changes that we were most interested in studying. In the outcome devaluation stage of the task, participants were told that certain outcomes were no longer valuable, so participants had to carry that information backward to ensure that they no longer responded to the stimuli that they learned led to that outcome. And in the stimulus evaluation stage of the task, participants were told that certain stimuli would no longer lead to valuable outcomes. So participants had to carry that information forward to ensure that they no longer responded to those stimuli. So to bring this more to life, in the first stage of the task, a participant might have to learn that a right button press is the correct response to a flamingo on the outside of a box, and that when that correct response is selected, a donkey outcome and points are presented. We found that women with bulimia nervosa and healthy controls learned, or in technical terms, acquired these instrumental discriminations at the same rate. And this was reflected both in accuracy, which is shown in this graph here, and in reaction times. We also checked and found that average accuracy in this stage of the task was unrelated to bulimic symptom severity. Subsequently, both groups performed at the same level in our outcome response knowledge test. They were equally able to press the key that was associated with a valuable outcome instead of the key that was associated with a now crossed out outcome. Together, these results suggest that individuals with bulimia nervosa and controls learned associations among stimuli, responses, and outcomes equally well. On the flips of action stage of the task, which assesses how people adapt to outcome devaluation, every 12 trials of this stage, participants were shown 2 new outcomes out of 6 that are no longer valuable. So in this case, they were shown 2 prize animals inside of boxes that would no longer lead to rewards or were no longer valuable. In the subsequent trials of this task, participants would then need to respond to stimuli that they learned led to still valuable outcomes, and they would need to exert control to withhold responses to stimuli that they learned led to no longer valuable outcomes. So for example, here in the example shown, the flamingo is a no-go because the previous outcome that it led to, which was the donkey, is no longer valuable. In the baseline test of the task, which assesses how people adapt to stimulus evaluation, every 12 trials, participants were shown 2 new stimuli, or in this case, animals on the outside of boxes, that no longer led to rewards. In the subsequent trials, participants would need to respond to stimuli that they were told could still lead to rewards, but exert control to withhold responses to stimuli that they learned no longer led to rewards. Importantly, this part of the task is different from a simple inhibitory task like a go-no-go task because it requires participants to repeatedly, over and over, update which of the 6 cues requires a no-go response and which requires a go response. They have to update this information and flexibly adapt 9 times total over the course of this stage. And they have to suppress previously learned stimulus response associations that they formed in earlier stages of this task and practiced in earlier stages of this task. So what did we find on these stages of the task? Well, we didn't find any evidence for group differences in responses to stimuli that led to devalued outcomes. However, women with bulimia nervosa responded significantly more to devalued trials on the stage of the task assessing for the ability to adapt to stimulus devaluation. And this reduced sensitivity to stimulus devaluation was associated with more frequent binge eating and more frequent self-induced vomiting behavior. To understand more about the underlying mechanism that could be driving this impaired stimulus evaluation, we used a computational model developed with our collaborator Vincenzo Fiore that estimated participants' trial-by-trial beliefs about stimulus response outcome associations, or Bayesian inference, and outcome response stimulus associations, or Bayesian inversion. In the final 2 stages of the task, the application of this model estimated on each trial and for each stimulus, each subject's estimated probability that responding or not responding would yield a rewarded outcome. We focused in on how these beliefs are updated according to Bayes' rule, similar to the Bayesian stop signal model that I showed you earlier, focused on updating beliefs about the need to inhibit responses. After each new presentation of new devalued stimuli or outcomes in the final 2 stages of the task, the no-go probability would increase for devalued outcomes or cues and decrease for valued outcomes or cues. In our model, how fast this adjustment occurred was allowed to vary per person, and the parameter that governed this pace of belief updating, so how quickly or slowly this updating occurs, is what we were ultimately interested in comparing between groups. In line with results from traditional analyses of behavior on the task, we found that groups did not differ on the computational parameter that indexes the pace of belief update during the outcome devaluation phase of the task. But the group with bulimia nervosa had a slower pace of belief updating during the stimulus evaluation phase. This offers us a potential interpretation of the behavior that we saw. So in the traditional analysis of the task data, we saw that when directly instructed to withhold learned responses to specific stimuli, women with bulimia nervosa were more vulnerable to errors. They had trouble flexibly adjusting to this change. The computational modeling results suggest that this aberrant behavior might be driven by maladaptive belief updating, which in bulimia nervosa resulted in a slower pace of behavioral adjustment when participants were presented with new information about the value of stimuli. We also found that this slower pace of belief updating was associated with more frequent binge eating and more frequent purging in the last 3 months. So why is it, do we think, that we only saw a group difference and only saw an association with symptoms when there was a change in the value of a stimulus and not a change in the value of the outcome? Well, there are some important differences in the instructions and the updating that's required on each of these 2 subtests of the task. The baseline test relies on standard Bayesian inference processes or stimulus response outcome chains that need to be updated depending on the new available information about the first element in the chain. On the other hand, because outcomes in the slips of action test are devalued by explicit instruction, this test requires updates via Bayesian inversion, or outcome to response to stimulus. Bulimia nervosa might be more strongly associated with problems updating beliefs that are more easily accessible, and we speculate perhaps closer to a classic habitual behavioral or automatic behavioral strategy. The cognitive demand and deliberation that's required in the slips of action test might be greater. And this idea was supported by slower reaction times on this phase of the task across all of our participants. Without slower, more deliberative processing engaged when outcomes are explicitly devalued, women with bulimia nervosa might have difficulty suppressing their learned and well-established stimulus response tendencies. These results support the idea that bulimia nervosa diagnosis and bulimia nervosa severity are associated with impaired flexible inhibition of previously learned stimulus response associations. And these results suggesting that individuals with this disorder might have a harder time adjusting to new information about cues or stimuli could explain why they seem to be more likely to get stuck in a number of initially reinforcing behaviors that have nothing to do with food or eating, from shoplifting to substance use. They could also have important implications for treatment. For example, longer versions of prolonged exposure to stimuli that can powerfully evoke binge eating and purging, paired with response prevention, might be helpful for individuals with more severe versions of bulimia nervosa. This is because that pace of belief updating or learning about strong stimulus response associations might be slower for these individuals. In addition, there are some data to suggest that repeatedly practicing inhibiting responses to certain stimuli actually implicitly devalues them. So perhaps practicing inhibiting responses to common cues for eating disorder behavior in neurocognitive training programs could be helpful. And perhaps interventions could capitalize even more on the outcome devaluation that we found to be intact in bulimia nervosa. This can include, for example, more explicit and detailed exercises to devalue eating disorder symptom outcomes and increase the explicit value of non-eating disorder related outcomes, such as improved relationships or graduating from school. To summarize, today I've shown you that after this internal state change introduced by eating, individuals with bulimia nervosa show a blunted neural signal for control-related expectation violations, and they show a decreased value of control-related cognitive effort. I've also shown you that after external contingency changes, in this case introduced by stimulus devaluation, individuals with bulimia nervosa show a slower pace of belief updating that might help explain some of the sticky behaviors that we see our patients get stuck in. So hopefully I've convinced you that prior accounts of bulimia nervosa as a disorder that's driven by impulsivity, dysregulation, and under control are incomplete, and that instead it seems more accurate and maybe more useful to consider it as a disorder that's driven by difficulties adaptively engaging specific elements of the control process. A continued focus on more precisely characterizing exactly how and when these alterations occur can help us develop and deliver more targeted interventions. Before I close, a few notes on research and treatment. If you're in the Manhattan area and are interested in taking part in our research, please reach out to us using the information listed on this slide. We're recruiting individuals really from the tri-state area who are able to attend visits in Manhattan. And if you're a researcher with structural and/or resting state scans of individuals with binge eating, please consider joining the Enigma Consortium. We're pooling data from groups across the globe to summarize alterations in brain structure and brain function in individuals with binge eating, so folks with bulimia nervosa, as well as individuals with binge eating disorder. We can only really start to definitively answer questions about symptom heterogeneity and really get more generalizable findings when we have very big data to work with. If you're looking for helpful resources to learn more about eating disorders and clinical care, the resources at the bottom of the slide have rich information for patients, families, clinicians, and researchers. Last, I'd like to acknowledge a host of collaborators and mentees across several institutions without whom this research and my current work would not be possible. And finally, my funding sources, particularly the Brain and Behavior Research Foundation. I'm very grateful that they've supported some of the ongoing work that I showed you today. Thank you very much for your attention.
Laura, thank you for an excellent presentation. You really explained how a lot of the more basic kind of work that you're doing could then relate to new clinical approaches.
Absolutely, thanks.
Let me ask you, where do you see this work going? If you were to present again to us in 2 years or 5 years or 10 years, where do you think it will go?
My hope is that we can really leverage some of these findings to develop state-specific interventions, which I kind of hinted at in some of my slides. We're currently doing work now that's focused on developing new neurofeedback-based interventions for folks with bulimia nervosa, where we can actually train individuals to change their own brain activation by showing them what's happening in their brain in real time and having them test out different strategies to find what works for them to change their activation. And we think, based on some of these findings, that it might be particularly important to have people learn these strategies and practice them when they're in very specific states so that their brain is primed and in the best possible state for them to get the most out of those interventions. So that's an example of one sort of direction forward and application of these findings that I see in the future. My hope is that with an eye towards precision medicine, we can not only be trying to figure out what interventions might work best for whom based on their history and symptom presentation, but also when might a specific intervention work best for that person.
Great. Well, I'm looking forward to getting that update from you down the road. Let me ask you, there's been a lot spoken about the fairly new medications that are being used for obesity. And I'm wondering if any of those have a role in these conditions.
That's a great question. We're certainly seeing that there are new problems sort of created for folks who struggle with binge type eating disorders, who might also have higher weight status and choose to start taking these medications, or might have comorbid diabetes and start taking some of these GLP-1 agonists. I think that's what you're talking about.
Yes.
And that can pose extra challenges for folks with eating disorders for sure, by again, sort of changing their experience of hunger and fullness cues, exacerbating some of the symptoms of the illness that they might already have. And so very careful screening for eating disorder symptoms is for sure recommended for anyone considering prescribing these types of medications to folks. But in terms of mechanism, I think there's a lot of work to be done both in preclinical, so for example, mouse or rodent models of binge type eating behavior, as well as in people, to try to figure out how it is that those medications or GLP-1 antagonism or agonism might affect control-related processes specifically. I think a lot of the work has focused, importantly, as it should, on reward-related processes and how motivations to seek out foods that might be particularly palatable or rewarding change, and how craving levels might change with these medications. But I don't think, to my knowledge, we know a lot about how these types of pharmacological agents might change control-related processes, like some of the things that I showed you today. And so I think there's a lot more to be discovered there and to learn there about what the effects of these medications really are.
Good, thank you. I want to ask you about, you had mentioned sometimes the comorbid conditions such as chemical dependency, certainly depression, etc. Could you
Yes.
speak a little bit about bulimia nervosa and some of those other conditions that sometimes happen along with it?
Yeah, so the most common comorbidity in folks with bulimia nervosa is major depressive disorder. And this is maybe not particularly surprising for folks who are familiar with these two diagnostic categories, in part because it turns out that one of the symptoms of major depression can be hyperphagia, which is just a fancy word to describe excessive eating or overeating behavior. And that's often induced by negative mood in folks with depression. We also know that depression can present with hypophagia, or changes in appetite, where folks find that they're just not as motivated to eat and might even experience weight loss related to their negative mood. But certainly seems interesting that we find an appetitive change that's associated with major depression as well. And it might then be no coincidence that for folks with bulimia nervosa, even if they don't have a comorbid diagnosis of major depression, often report that their binge eating and purging episodes might seem to be kind of kicked off by increases in negative affect. So in both of these conditions, we tend to see an association between a negative mood state and a tendency to binge eat and purge. And actually some of the work that's led by my postdoctoral fellow, Blair Shevlin, and with collaborators at Columbia University, including Joanna Steinglass and Lauren Giannini and Karin Ferd, have really tried to understand how it is that these changes in negative affect or inductions in negative mood might actually change the way that people with bulimia nervosa make decisions about food or choices about food. So that's one really common comorbidity that we see with folks with bulimia nervosa. We also tend to see, as you mentioned, higher rates of other sort of disorders characterized by problems with some form of self-regulatory control. So as you might guess, based on that statement, we see higher rates of substance use disorders. Those behaviors can often feel out of control to individuals. We also see higher rates of ADHD, interestingly, in folks with bulimia nervosa, and we're trying to understand more about why that is. There are common sort of brain-related substrates contributing to both symptoms of ADHD and bulimia nervosa. We also see relatively high rates of a history of some sort of trauma in the past, and in a lot of cases, full-blown PTSD in individuals with the disorder. And there's some ongoing work trying to understand why that is from a neurobiological perspective. How might that trauma and symptoms of PTSD affect brain circuitry in a way that might make folks more vulnerable to developing binge eating and purging symptoms? So those are some of the comorbidities that we see that sort of seem related to behavioral discontrol. We also see a relatively high comorbidity with anxiety disorders in general that are, just have an elevated prevalence in the population. So generalized anxiety disorder, as well as other anxiety disorders. And we can think of those as in a way as sort of under controlled cognitive symptoms of having anxiety and feeling like it's difficult to stop once you kind of get on a mental track of maybe worrying about the future. You could think of that as sort of a difficulty exerting control as well. And there are some data to suggest that we see somewhat similar alterations in the brains of people who only have anxiety disorders that resemble what we see in the brains of people with bulimia nervosa.
Good. Well, Laura, I want to thank you for joining us today, and more importantly, for the ongoing work that you've done and will continue to do on this important field. So thank you very, very much. I
Thank you.
also
Thanks so much for having me.
Thanks. I also want to thank everybody who joined us today. And I'd like to ask people to please consider making a donation to BBRF today. 100% of every dollar donated for research is invested in our research grants. We're able to do this because our operating expenses are covered by separate foundation grants. This means that when you donate a dollar for research, that dollar goes directly to the scientists. To make a gift, please visit bbr.foundation.org or call 1-800-829-8289. This webinar has been recorded. If you've missed any portion or would like to share it, please visit the events and webinar page on our website. And please join us again next month on September 10th at 2 p.m. Eastern Time when Dr. Jenna Trainer, a clinician scientist at McLean Hospital, will present new diagnostic tools to predict symptom improvements in personality disorders. Thank you, and remember, together we can dramatically improve the lives of those living with mental illness and enable more people to live full, happy, and productive lives. Thank you.